be as long because you get the reusable face mask application that you can put on with cloth, take off. But I'm assuming this is an application that it can go into?
Yeah, yes, because actually this extract can be used in the face mask and the effect it's the best compared to other cosmetic products like serum or cream, because when it is in the mask, the tightening effect can be stronger and play the best by this extract. And just like you can add 0.1, 0.5% to 1%, it's enough for the face mask. And also, if you want to use in other products like serum or cream, it is also okay. And just increase the dosage a little bit higher.
Okay, great. Yes, because we see a lot in the industry with, especially with face masks. So, especially the instant gratification, instant tightening, and a lot of these Hollywood superstars before they go to an event, they actually, a lot of people use these on their face. So that would be an extra application other than just like you say in a serum or a cream or something like that. So, Komotso, are there any questions on your side?
Just maybe one. Lani, has this been tested in comparison with any ingredients? I know you mentioned, like, when you were talking about the structures, you mentioned also hyaluronic acid. Has there been any studies comparing the firming effect with such ingredients?
Yeah, thank you for the question. And I think it's a question commonly asked by many other customers also, and I would like to explain it. So firstly, the structure is different from hyaluronic acid and like beta glucan. So we know hyaluronic acid is very good for moisturizing, but it can hardly have such anti-wrinkles or lifting effect like the peach gum extract because of the structure or some other extraction technique. And for this Prunus persicum gum extract, we do compare with the benchmark. So we know that there are many instant anti-aging, instant anti-wrinkle, or the lifting product in the market. So we compare with those ingredients. And finally, it's not very convenient to say what products we compare, but we can explain the result. So compared to all the actives in the market, so if we talk about the instant anti-wrinkles actives, our extract, it's the greenest. It has the highest ISO 16128 natural index. So it is the most natural one. And if you compare with the natural instant anti-wrinkle actives, this peach gum extract, has the most effective. It is the most effective one among the natural. Yeah.
Thank you so much for that. And just maybe to just add on a little bit on that, I understand that when you apply it, it will give you that instant, feel and the instant firming and lifting. Have you maybe seen, does the skin get better with time? Like, will your wrinkles just be reduced or your skin firmer just only on application or even probably, the next day? Do you see any better improvement in your skin with repeated use? For example, let's say
Yes,
I'm
yes.
using it this week and then next week I'm not applying it for a day. Will I see a better appearance on my skin?
Yeah, this is also a very good question. So we believe that different products, different actives, they have different position. So for this extract, the position is for instant anti-wrinkles that can bring consumer confidence and they can feel the effect instantly. And in order to strengthen and improve the skin condition, we will recommend to combine with other long-term anti-wrinkle products. For example, we have one ingredient, it's based on the oligopeptide. After the combination with long-term and the short-term, we can really improve the skin in the long term. Even the consumers will stop using these products. So on the other hand, because this gum extract it's from polysaccharide, we know the polysaccharide has very good moisturizing effect, and the moisturizing it's good for anti-aging. So we can say that even we the customers don't use it, if you use it for a long time, it can also improve your skin condition.
Awesome. Thank you. That's
Perfect.
that
Thank you,
from my
Lenny, for
side.
that answer. Yes. So, well, we'll have to put you to the test. So, if you are speaking again at COSCEM, and definitely even in 5 years' time, if we're seeing that beautiful glowing porcelain skin, which we're seeing from here, if it's still looking the same, we know your product is working very well because your skin looks like porcelain
you.
from this side. So, thank you for sharing that. It's the ingredient looks lovely. Thank you for the presentation. It sounds lovely coming from the jade. Is it the Jade Dragon Mountain? It sounds very
Yes.
exotic. I'm liking it already.
Yeah, Jade Dragon Snow Mountain.
Yeah, that sounds amazing. So thank you for being part of the COSCEM conference. It's lovely to hear all about this new technology coming out with instant tightening and some instant gratification in skincare. So thank you, Lani, for joining us.
Thank you,
Thank
Lenny.
you. Thank you.
Thank
Have a nice
you.
day.
So we're going to actually move on to our second last speaker for the session, and I always find it particularly interesting in the baby care, baby arena, because anything that's happening in that is always very sensitive,
Yes.
and any new information and new technology. So even with regards to what's happening in baby care with Johnson & Johnson and talc being taken off the market as such, we always sort of keep our ears open for any issues and any new development in baby care, and especially microbiome friendly baby care products. So we're going to be introducing our next speaker online, who is online waiting, and I hope I'm pronouncing your name right, Leo.
Right, yeah.
I don't want to
Correct.
say Eeyore, it's Leo.
Well, you can. I will answer anyway.
So the only thing I know is shalom. So, and yeah, thank you for being part of today. It's wonderful to have
My
some
pleasure.
representation from Israel, very special country. So we'd like to welcome you today to speak, and you're also on our discussion and debate panel later. So we're also welcoming your advice and your recommendations there. So you're going to be getting a lot of COSCEM today, and we're going to be getting a lot of you. So
Great.
if you are ready, you can start your presentation. Thank you, Leo.
My pleasure. So today I will talk about, let's say, preservation from a different perspective, maybe things which are more popular nowadays, things which are related to clinical, or let's say things which are suitable for baby skin and sensitive skin. This is, of course, connected to many things, also to the pandemic that we had. Can you see the second slide, by the way? I mean, like it's going on, right? I assume yes. So the sensitive skin issue is something that is actually discussed for many years, I would say. Like I would say the last 15 years, it became a more and more subject which everybody discuss, and it's also connected to preservation. Obviously, the sensitive skin is a global issue, and due to the pandemic, it even became something which was more relevant due to the fact that skin was exposed to rinse off and leave on product in a much higher level. And we saw that it's a global thing, which means it's happened worldwide. You can see it in the US, in South and Central America, in Europe, and also in East Europe, and also in Asia. So you can see here the ratio and the percentage supposedly difference, but then again, it's a concern which became bigger. Also, sensitivity is connected to the usage way, because eventually when you use cosmetic product, it can be applied and remain like face cream, wipes, or makeup, which means actually you apply it on the skin and it stays there. So it needs to be sensitive enough due to the fact that it stays on your skin. While, for example, there is also the option of reapplication, which means using it again and again during the day due to the fact that you need it, like lipstick, hand lotion, and so on. Another aspect of the sensitivity is also the area, because you can talk about many areas on your body which are more sensitive. We talk about the area on your face, the mucous membrane, for example, or intimate area, and also areas which are connected to sensitive skin from the beginning, like newborn or baby skin. So we talk about products which need to be applied to this kind of product and need to be sensitive originally. Obviously, the pandemic, as I mentioned before, contributed to this also worldwide. And we saw that the response to the issue of irritation were actually increased. People were more reacting to using a cosmetic product and referring to the fact that it should be more, let's say, sensitive to the skin itself. We get higher claims from customers worldwide, which talk about drier skin due to the fact that you use more rinse-off product, the skin rash issue, and the itching issue when you deal with a product which are less sensitive to the skin itself. And of course, when we talk about sensitive skin, it goes up to 28% due to the excessive washing, the hand disinfectation, and the mask wearing. Now, the mask wearing, for example, it's of course related more to the face. And due to the fact that people used a lot of the masks on the face, it means that they were exposed more to the rash and irritation issue. Now, when we're addressing sensitive skin when preserving formula, obviously it's something which is connected to the formula. It's not necessarily means that all irritation, sensitization issues are connected to preservation, of course. But then again, the preservative should be aligned with the marketing trend that the final product is aiming for. And what we saw, that whenever we talk about sensitive skin, people are talking about free-from option, which means avoiding things which are connected to the irritation sensitization factor. They talk about also about dermatological tested, which means actually this was tested and proven to be sensitive enough for the skin. Also, the fact that you want to use as low as possible. And this is relating to the preservative specifically, which means you need to use as much as possible, but it should be low in order to not create or, let's say, contribute to the issue of the sensitization and irritation, which means we are aiming for more innovative technology which should be aligned with all this, or what we call smart preservation, something which will give the right dosage of preservation, but not contribute to the irritation, of course, negative contribution. So what we did in Sharon, we developed during the COVID, and we had a lot of time back then. I mean, like you had 2 years of working, and we developed actually a wet wipe line, which was actually trying to focus on this sensitization issue, talking about sensitive skin, generally speaking, for babies and newborn, because generally speaking, when we talk about wet wipes, it should be more directed to this direction. And obviously, when you deal with wet wipes, it's considered to be the most difficult, let's say, application to preserve, because you have the wipe, you have the lotion inside, and the wipe is actually in the lotion, and you have the package, which means, and you put the finger inside all the time, you take the wipe out, so you actually continue to contaminate the product constantly during the usage, the shelf life. So it needs to be preserved properly, which means preventing the contamination, and still need to be relatively good for the sensitive skin, which means not exposing the baby or the newborn or the sensitive skin area to sensitization and irritation. So why preserving wipes is so challenging? Well, basically it's connected to the formula itself. And I mentioned before the wipe, the lotion, and the package. The wipe can be contained from many fibers option. Typically, it's combined from viscose and PET, but it can be also bamboo, hemp, paper pulp, and so on. And also the widths can be different. So the fiber is one thing. Also the lotion, the formula itself is actually contributing to the final product, which means it can bring all kinds of ingredients to the fabric, which you actually put on the skin. So the combination of those two are actually what's left on your skin after cleaning the area which you want to clean, which means it stays on your skin, and then it should be, I would say, sensitive enough for the skin area. So let's say that from our perspective and what we see during the last, I would say, 15 or 20 years, that the direction of the wet wipe specifically was more into the inky, which is more clean or free from. Because maybe 20 years ago, you could see that still parabens and isothiazolinones, which are classic preservatives, are used in wet wipes. But what we saw during the, let's say, from 20 years ago and forward, people wanted to get away from the classic preservative and look for other options which will be more directed to the free-form option, which means not containing the classic preservative, but also cleaner inky, or what we call pronounceable inky, things that the customer can read and understand supposedly what he's using. So then classic preservative are out of the question, and using things which are avoiding like things like parabens, phenoxyethanol sometimes, and also alcohol. I would say that phenoxyethanol is the current trend, which started in France like 8 years ago, and it came also to the wet wipe segment. And it's, of course, it's only marketing claim, it's not legislative. Then again, this is one of the current free-from option. While from the other perspective, new claim emerged due to the COVID, but also due to other perspectives which are connected to healthy lifestyle and awareness of the customer to try and use less chemical, in a way, names. So trying to use eco-friendly and sustainable ingredient, try to use formulation which were dermatologically tested and proven to be safe for the usage on the client, which can be a baby or a sensitive skin area. And also the last perspective is the biodegradability, which is kind of a new claim. Obviously, in some part of the world, it was never new, because in Scandinavia, for example, it was always something that people were concerned of, the way that actually we treat Mother Earth. But nowadays, I would say that biodegradability became something which is people are discussing worldwide. How actually the final product will be actually connected to Mother Earth, and if it will be biodegradable or not. So in wet wipe, we talk about the wipe, and we talk about the lotion. So it's complicated. So what we try to do with the smart preservation idea, which I mentioned before, is to find actually, let's say, the right preservative for this specific difficult product to preserve, which will actually prevent the contamination in the right place. The right place is actually the lotion and the wipe, right? The package, what actually inside the package. And obviously, we talk about minimum quantity possible because we think that we need to use preservative according to the need. It's not the right thing to do to add too much because it will not do anything. Eventually, preservative should prevent the contamination during the shelf life that you actually claim on the product, which means after opening, you need the preservative to keep the product safe until the product is finished, which means the shelf life. So what we found when we did our research about wet wipes, we found that actually there is an issue of biofilm which can be created on the fiber, which means biofilm is kind of a way of the bacteria to connect to surface and to grow on it. And actually by that, containing some kind of contamination, which is very difficult to get rid of later on. So avoiding the biofilm is something that everybody desires. It can happen when generally speaking about products. When you produce product, it can grow on the wall, on the floor, in the machinery. But when we talk about the final product, it can, in wet wipe, for example, it can grow on the fiber itself. And here you can see an example of a microscopic picture on the viscose fiber part of the wet wipe. And you can see here Pseudomonas aeruginosa, which is a gram-negative bacteria, grows on the viscose itself. You can see it also by E. coli, another gram-negative factor, which grow on the viscose. And also, for example, gram-positive like Staphylococcus aureus, which can grow on the viscose. So we found that the viscose is actually connected to the biofilm growth in wet wipe when we talk about final product of wet wipes. So what we wanted to find is actually a way how to avoid the growth of the biofilm on the fiber, which means preventing the growth of the biofilm on the viscose part of the fiber, because typically, as I mentioned before, it's a combination of viscose and other options, PET, bamboo, hemp, paper pulp, and so on. So to find a preservative system which will deal with the biofilm and obviously the lotion itself. What we saw that most preservative, when we talk about PET and viscose, there is an issue of hydrophobic and hydrophilic. And when we talk about preservative, typical one, which are old school preservative like parabens, or for example, organic acids which are used nowadays, or boosters, or like ethylhexaglycerin or phenoxyethanol, most of them are hydrophobic. While when we talk about preservative, when we talk about things that we want to deal with viscose, actually we want it to stay, which means actually stay and take care of the viscose issue. We found that actually most of them will actually will abandon their position as a preservative and will not take care of the biofilm growth. So what we try to do is to find a way how to combine an hydrophilic preservative and not hydrophobic, which means we'll stick to the viscose and take care of it, and also an anti-biofilm agent, which will deal with the biofilm issue of the viscose. And by that, creating a preservative solution which will deal with all the aspects of preservation in wet wipe. So taking care of the lotion and the fiber, and by that, having a good product which is protected during the shelf life. We found during the research that we did that maltol, which we have in other line, can be a good anti-biofilm agent. By using maltol, which is a food additive under the name E636, which is actually taking the position of from food to cosmetics, because maltol exists in food, we take it to cosmetics. It's a very nice ingredient which can work as an anti-biofilm agent, as well as a chelator and also some kind of antimicrobial agent. With a very good water solubility. It's a white powder with a light caramel, like cotton candy smell in a way, like very light one. It's grass and exists in nature, so we have it from both options, from synthetic source or from natural source. And it's also readily biodegradable. So it's we have a lot of nice claims about the maltol. And the fact that it's an anti-biofilm agent, it's actually the fact that actually helped us to select it for this slide. We found, and you can see here the reduction of the biofilm growth on the fiber. You can see the bacteria which we choose, like E. coli or Aspergillus brasilianus, which are, one of them is gram-negative and one of them is fungi, mold. You can see the maltol is actually helping on both cases, which mean keeping it safe. To complete the system, because as you probably know, there is no one ingredient which can give full broad spectrum protection of preservation. Broad spectrum preservation means gram-negative, gram-positive, and the fungi, the yeast and the mold. So in order to create a system which is broad spectrum, you need to combine elements. So we took the maltol, as you saw in the last slide, and we combine it with zinc species. We chose specifically zinc acetate, which is also a food additive ingredient, E650. And also it's water-soluble hydrophilic ingredient with which coming as a white powder, GRAS approved, readily biodegradable, and very high limitation as is. So we used much less than what is mentioned here. So very low level of zinc acetate combined with maltol in order to create the system that I mentioned, a full broad spectrum system which will deal with the wet wipe aspect of preservation. Here you can see an example of challenge test that we made in 0.7% when we combine the maltol with zinc acetate and combine with sodium benzoate, because organic acid or organic acid salts are actually some kind of an anchor in wet wipes. Typically, if you will check worldwide, you will see that nowadays most of the wet wipes contain some organic acid in order to complete the preservation aspect of wet wipe. So the sodium benzoate choosing or organic acid, generally speaking, are a given thing. We took it as the anchor of the system. We combine it with zinc acetate and maltol in order to create the full system, or you can see later on we create also other system without. So we have both options. When using 0.7 of this system, which you see below, you can have 0.7 was good enough to pass challenge tests according to the EP and USP criteria, because you have the anti-biofilm agent, the hydrophilic system, and you gain a very clean and short inky in order to gain all this. Now, the pH that we used was 5.5, and this is very typical to wet wipes. Acidic pH is something which is normally used in wet wipe. If the pH will be lower, you can use even less of the preservative system. And typically, the pH is lower. And when we talk about wet wipe, we talk about acidic pH, which means the squeezed wipe. It's not the lotion and it's not the fiber, it's the combination of both. Because typically the fiber will be basic and then it takes the pH a bit up. So in order to understand what's the final pH, you need to squeeze the wipe and check the lotion which coming out of the fabric. So what we created is actually a full protection formula for the non-woven or any fiber that you have there with all kind of combination. You have a very high efficacy and low level of use, broad spectrum protection, and the anti-biofilm mechanism. The safety and the inky is very nice and very friendly. And of course, maybe I didn't mention before, it's free of solvent because it's all powder. So the powders are solubilized in the water phase, which is, very big phase in the wet wipe. So obviously it's free of phenoxyethanol as well. So this is a solution which is a scientific breakthrough due to the anti-biofilm mechanism and also giving a broad spectrum solution to the final product, which is also clinically tested, readily biodegradable and microbiome friendly, which I will talk a bit about later on. So what we did last year and this year was actually taking this idea into the leave on and rinse off. And this is something that actually we launched during exhibition this year, the combination of this anti-biofilm agent, antibacterial agent with the zinc acetate and sodium benzoate that took it to the next level. Just to show you a bit about the activity of maltol from microscopic perspective, how it actually works on the Candida albicans or Aspergillus barzilianis, the fungi. You can see that by using the maltol, by using TEM system, you can see that there is a membrane damage and then intercellular material which is losing, and then you actually inhibit the growth of the contamination that we desire to prevent of the growth. So actually we took it to the next level, as I mentioned, and we took it to leave on and rinse off, and used natural maltol here in order to create a system which is working with sorbic acid and sodium benzoate. This system is Cosmos approved, which means it's natural according to the Cosmos standard. So by using natural maltol, what you saw in the wet wipe, we use synthetic maltol due to the fact that typically wet wipe are cheaper. But if we take it to leave on and rinse off direction, which is more Cosmos and natural oriented, we typically would like to use as much of natural ingredient as possible. We use here natural maltol combined with sorbic acid and sodium benzoate, which are acceptable synthetic preservative from Cosmos perspective. So you have actually a system which is based on natural approach, friendly and safe, short inky, vegan, obviously, all our products are vegan, but it's a claim. Again, all ingredients are actually have an E number, so it's actually the approach of from food to cosmetics, and it's also clinically tested and readily biodegradable, this combination. So you gain all the perspective which are needed for sensitive skin in one solution. Showing you a bit of the activity, when using 1%, again, the pH, the upper pH is 5.5, due to the fact that we use organic acid or organic acid salt. The lower you will go with the pH, you can gain actually more activity, and then maybe you can use lower level of preservative. Here we used 1% for basic cream or for shampoo, and you can see that it's passing after 28 days the EP and USP criteria of challenge test. Another option that we chose was the combination of zinc acetate and sodium benzoate. This is also another option of from food to cosmetic approach. Both have E numbers, so it's a minimalistic patented idea which is combined from this aspect, combining these two products in acidic pH in order to create a free-from vegan option to use in leave-on and rinse off. This was also used for wet wipes, but we took it now to the leave-on and rinse off idea. So it's a very short and friendly and smart preservation from the approach of the from food to cosmetics is again relevant here. And also this combination was clinically tested and microbiome friendly approved, which I will show you in the next slide. And also from an environmental perspective, it's readily biodegradable. So here you can see an example of challenge test we created with the shampoo. Using 0.7% of this combination was passing challenge test according to the EP and USP criteria as well. Now I will show you a bit about the safety and clinical tests that we did. We of course wanted to cover all aspects of tests in order to prove that it's non-irritant. So we checked patch test on sensitive skin and the potential irritancy on ocular, and on the continuous usage level, and we found that in the upper level we recommend, it's non-irritating. If someone will be interested to get the full documentation or the report, it's available, of course, from our side. But what we actually proved, it's not irritating in all areas of the body, and also for sensitive skin or newborn and baby. And this take me to the next level, because we also proved that this option is also microbiome friendly by checking actually the balance of the negative and positive microbiome on the skin. We found that it's not harmful for the microbiome species, which means keeping the good stuff on the skin and expanding the bad stuff. So it's actually not eliminating all bacteria, but actually some kind of selective, which means keeping the good balance on the skin. So I talked a bit about the options that we can offer regarding preservation, which are more in the newer side of approach, sensitive skin, safer to the body, the impact on the skin, which means sensitization, irritation, but also microbiome friendly. You can gain all this by the combination that I mentioned so far. That's it. Thank you.
Thank you, Leo. Your presentation was so thorough.
That's what I thought.
Covering like every aspect. Okay, so this looks like too good to be true, almost in a sense. So one of our main questions is, would there be a challenge? What would be the biggest challenge then for your manufacturers manufacturing your baby care products to actually move over to this this type of formulation or this type of ingredient in their technology? What do you perceive as the biggest challenge for manufacturers to move to this?
Well, I think that it's all about marketing eventually, because whenever there is a marketing demand to move to this direction, then the R&D need to find a way how to deal with this perspective of looking for preservative which will be less irritative, more relevant to sensitive areas, and dealing with the free-off aspect, the blacklist supposedly, of what the marketing demands are nowadays. So it's a process, because you need to implement it in the R&D products. I mean, like the new launches, you need to put in the formulation and see the compatibility, the stability, the challenge test. It's a process which can take a few months, I would say. And of course, there is a reason why you need to go for that. I mean, like, I would not change preservative system unless I have a demand from the marketing or from the market, of course, because this is the most difficult thing to do. It's changing the whole aspect of the formula. So if you have a demand and you need to follow this demand, you need to find the right preservative to follow this demand. That's it, to my opinion.
So did you, well answered, thank you. So did you say, because I've made some notes, but now I can't read my own writing, did you say it was used at 0.7% or 1% in your presentation?
Well, the first option that I showed you, the combination of the natural maltol, the natural direction, natural maltol with sorbic acid and sodium benzoate, this one is up to 1% typically. The lower you go the pH, it can be even lower level. While using the sodium benzoate and zinc acetate combination, then you can go up to 0.7 and can have actually a full broad spectrum solution for rinse off and leave on.
So then really it's comparative dosage wise to a phenoxyethanol or something that's standardly in the market, but then is there a very big differential in cost comparison? Because like you say, it's about the demand in the in the world for these type of products. So if the demand is there, or if it's not there, is there a big difference in cost with regards to this, or is this comparative also with regards to dosage? Is cost-wise, is it one of those easy things to just transfer from one ingredient to the next with regards to cost?
So it's a good question, because eventually the comparison should be according to what you would try to gain. Comparing this, for example, to classic preservative like parabens, isothiazolinone, formaldehydanones is irrelevant, because those are actually used in a very low level and they are cheaper. If you want to compare something, then better to compare apple to apple, which means compare this option to other natural preservative
Yes.
which exist in the market. So if we do that, it's a cheap option, because our solution, to my opinion, and as I saw, because I give the global technical support from Sharon, and I see that from pricing perspective, it should be very competitive, both product which I showed.
Okay, well, good answer. Thank you. Because often that's the challenge is that sometimes there is a demand, but then it's a cost thing. And then are you comparing apples with apples?
Exactly.
So it's, it's always wonderful to, to be exposed to all of this. And we understand definitely with the bigger corporates, the lead time in changing, and especially, it's the same as sunscreens too. Sunscreens and preservatives are those things that you really don't change unless you have to and unless there's a major push from the market, and also obviously
Exactly.
your cost.
And I can mention another thing. I mean, like since our solution was based on wet wipe from the beginning, wet wipe is a very cheap product. So from the beginning, our solution was aimed for wet wipe, and when we took it to the next step, we kept actually this direction of pricing. But when we took it from synthetic maltol to natural maltol, obviously it will be a bit more expensive, but relevant. I mean, like if you compare it to existing product in the market, it should be much more cost effective, I would say. Cost effective is also relating of course to percentage the level of use. So cost in use is actually the right term in order to understand if it's relevant from pricing or not. And if you mention UV product, I have to mention that maltol was also found to be very good for a high UV filter product, because the polarity index of the maltol allow it to stay in the water phase and then protect the water from contamination, which happen a lot during when using preservative which are not water-based. Typically, most of them tend to migrate to the oily phase and then actually expose the water phase to contamination of gram-negative. So when using maltol, like the option which I showed you as well, can be also good for UV filter product and be cost-effective.
Oh, that's great information to know, because I also found it interesting. I didn't know this. I don't know a lot of things. I didn't know that when you squeeze the wet wipe, that's
None
the
of us.
pH. That's the pH that you take. So thank you for for explaining a lot of things, because not everyone is preservative or microbial experts. So that was something great. And then also, I'm assuming this product is patented.
Yes.
Yes. Okay. Perfect.
It's a patented idea, and actually we have this combination of maltol with polycretinium 80 or didecyl ammonium chloride, 2-cationic surfactant. So if you want to avoid the pH issue, which is combined with, connected, to the sodium benzoate or benzoic acid or sorbic acid, you can use maltol with this cationic surfactant and have a solution which is non-pH dependable, which means 3 to 9 pH with no problem. This is relating, of course, to UV filter product, if you want to avoid the acidic pH issue.
Oh, Leo, that's
But
great.
we are here
Great.
to answer questions. I mean, like if there are questions later on from customers or anybody, I will be happy to help with the formulation help.
No, thank you very much, because there's lots of additional information that you've given to us today, also with regards to the sunscreens. And it's discussions like this that bring it out, and we're going to see you a bit later today. So, so we're going to discuss more. So definitely, I'm sure there'll be some more questions as well. So yeah, we
Sure.
thank you greatly. Like I say again, special place of the world. Shalom, and we'll see you after the lunch break. Thank you for
Thank
being
you.
part of COSCHEM this morning. All right.
Great. Bye-bye.
Thank you.
Bye.
Bye.
So moving on to the next part of the world. We're now in Italy.
We're traveling the world today.
We're traveling the world today. And we have a presenter from Givaudan. And I hope I'm pronouncing the presenter's name, Giada?
Correct.
Correct. I love it. That's such a pretty name. Thank you for being part of the COSCHEM conference today. And we're looking very forward to your presentation. We love any new information, especially on, botanical extracts with any pharmaceutical-like extra mechanisms. So we are looking forward for you to share some of the technology and any efficacy studies that you have today. So you're welcome to start. Thank you.
Okay, so thank you, everyone. I am actually very honored of having the chance to present this brand of actually purified molecules from Centella asiatica that have been validated for effective skin and extracellular matrix repair. And I am understanding that you are really literally touring the world today, and I'm really delighted and honored of being part of this world tour on influential beauty. So thank you again. So we shall start actually from the plant. We discussed that it's a botanical extract. So it has a botanical source. And the botanical source is Centella asiatica, which is one of the most known, I would say also used and valued medicinal plants. It has many uses and traditions around the world, and as we will also see, many names too. So this plant that you're seeing here is Centella asiatica. It is a leaf. Actually, it may pop up very big on your screen as it does on my screen, but this leaf is actually the size of a small cherry. So Centella grows aside of cultivated fields, so it grows spontaneously in Madagascar and in many other Asian or African countries. Nevertheless, the Madagascar quality is recognized as the best and also the most consistent quality. So 2022 is actually in the Chinese calendar the year of the tiger, and it is with the tigers that we can start talking about the properties of this plant. In fact, it is actually common knowledge that when tigers were more numerous and not actually an endangered species, and they used to fight, as tigers normally do, for territory, over females and all of that, over food, they used to roll in centella grass to rub their wounds on the centella grass against it. So we know that now. Why? We know that because centella actually helped the animals to heal from those wounds. So in some way, to some extent, we shall attribute, I would say, the very early observations of the medicinal properties of centella to tigers, to these majestic animals. And this is somehow recognized in the fact that one of the many common names of this traditional plant is actually tiger grass. It is also claimed that centella was already used as a panacea in China over 3,000 years back. So the plant is known by many names. It is known as Centella asiatica, even though it's kind of weird because despite the, I would say, adjective asiatica, the large majority of this plant for medicinal uses is actually coming from Madagascar, so from Africa. It is also known as Indian or Asian pennyworth, Hydrocotyl asiatica, or Gotu-kola. And Gotu-kola, for instance, in Sanskrit means fountain of youth. Then there is also a name in the language of consumers which knows Centella asiatica as chica. So chica creams are actually seeing a massive growth, especially in the Asian countries. So despite the many names, actually the centella that we are talking about has a very fixed composition. So when we talk about natural ingredients, we tend to consider, I would say, a certain level of variability. Well, that is not the case, because in this case, the Centella asiatica is obtained by a very high purification fractionation of 3 molecules that are then recombined together. So we see the composition here. Centella asiatica, the Centella asiatica we're talking about, contains 40% of pure asiaticoside and 60% of the blend of the 2 acids, madacic acid and asiatic acid. And that comprises 100% of the product itself. So the sourcing, I already mentioned it, is from Madagascar, where the quality of the leaves is higher and more constant. Here in Madagascar, our suppliers are UEBT members. So, of course, traceability is assured, even though it's a spontaneous collection. So traceability is based on the villages and the areas of collection, and also the good formation of the pickers on plant conservation is assured. So pickers know that they should only collect the leaves and not pull off the whole plant with the roots. So we are also developing with the supplier and UABT additional sustainability activities that are related to social aspects. And as of now, community programs, especially on schooling, are ongoing in the villages and in the regions that are specifically dedicated to centella harvesting. So we have seen that centella is actually known by many names, but it not only this, because it is also considered for many uses. So you might already know Centella asiatica definitely in your market. Yet I would say it is an extremely versatile active ingredient, and it is used in very many cosmetic domains. For instance, according to the different markets, there might be a different prevalence of use. As an example, in 2021, it has been identified as the mainstream ingredient in China in products for sensitive skin. Or still in China, centella is much used in oral care products. So 42% of Chinese consumers would like to have centella in their oral care products for, in a way, more advanced and more active oral care product. In other markets, for instance, like in Europe, it is very much used mainly in body care applications, whereas my understanding is that in the African market, it is much used for soothing and skin repair, which is, as we have seen for tigers, the very, very root of the origin of the efficacy of Centella asiatica. So as in many cases at Givaudan, we normally start with transcriptomics data that normally indicate on what genes and to what extent our centella would be capable of giving an effect. So the question here is centella is able to induce the overexpression or the down expression of some specific genes that are involved in skin repair. And specifically, in this case, we looked at the relevance to the stretch mark biological formation. Just a little bit of introduction on stretch mark. A stretch mark occurs to our skin when our body or our skin is stretched too fast in too short a time, and the dermis underneath cannot keep up with the production of the and the organization, especially and the organization of the relevant fibers of the extracellular matrix. That is when a stretch mark occurs, and a stretch mark is just as a lesion, as a wound, without having the real wound. So it's a wound in the dermis. So we have been considering the markers, some of the markers of stretch marks for skin repair. And if we look at the cellular level, stretch marks are very similar to fibrosis, especially in the way the extracellular matrix is disorganized. So our focus in this case has been on mainly on genes that are expressing a status of fibrosis and on genes that are normally disrupting and cutting down the extracellular matrix components. So we can see here that the genes associated with fibrosis are overexpressed in fibroblasts at the stretch mark level, but treating with centella, they are no longer overexpressed. And similarly, this happened with the catabolic enzymes with the MMP1, which is actually the most relevant, as well as with the MMP7. So actually, centella is able to induce a major protection on the expression of catabolic enzymes and also on fibrosis markers. And this is, I would say, the first indication on how effective centella can be on skin repair. So following to transcriptomics, we carried on some biological evaluations on skin repair. And of course, I would say the most common and the most applicable study on skin repair is the scratch test. So as we consider mainly stretch mark is a lesion in the dermis, we wanted to mimic this with a scratch test by mimicking a lesion in the keratinocytes confluence. So what happens is keratinocytes are actually grown in a petri dish, and then they are mechanically scratched, mimicking the wound, mimicking the lesion. And then what happens is that we followed and observed how fast and with what organization, with what structure the keratinocytes were able to get back together and somehow to close the scratch, to fill in the scratch. So here's what we are seeing. I know the graph is quite complex, but I will try to do my best to drive you through. So if we look at the control, I'm sorry, I'm trying to indicate, but I cannot indicate. So let's just mention it. The light blue line is the control line. Then we have the positive benchmark, which is a fetal bovine serum used at 10%, and that is the line below the dark blue bubble. Everything else, all the lines that are on top of these are the different dosages of Centella asiatica. And if we look at the dosages, these were very, very little, very, I would say, feeble. So we had dosages ranging from 1 to 25 micrograms per milliliter, which would be, we see that on the bottom part of the slide, 0.301%. That is very, very limited. So actually this graph is telling us that Centella asiatica builds and promotes the cellular confluence, so builds and promotes skin repair. And also the second message that we are getting is that in this test, in this assay, the efficacy was not dose dependent, because on the different dosages we got fairly very similar results. So here's the, I would say, visual representation of the graphics that we have just seen. So the control at 24 hours is still separated, but not so the positive benchmark, which is, again, the fetal bovine serum, which has reached full confluence. And we can see that at extremely low dosages, the keratinocytes have been reaching full confluence as well. Then a second biological evaluation, this time on ex vivo skin explants, was carried on with different endpoints. So we have evaluated the capacity of Centella to promote collagen organization and also to fill in fibers occupation. We'll see what exactly it means. So when a stretch mark is formed at the dermis level, we have a disruption in the dermis network with the fibers of collagen that are altered, and their organization is also altered. And we have some empty spaces that are somehow filled in by immature fibers. So the collagen, in a way, fails to organize itself in bundles, and these immature fibers are actually filling the space, but they are not efficient on the, I would say, structure function side. So actually the stretch mark forms not because a real or massive lack of collagen, but because of a lack of mature and organized collagen, because of a lack of collagen organization in the fibers. So in this ex vivo study, skin explants from a 60 years old donor with stretch marks, so the explant did have stretch mark in the explant itself, were treated topically with Centella at 0.5% for 5 days. Of course, the dosages between cells culture and ex vivo and in vivo studies need to be different, but yet again, we are at quite a low dosage of 0.5%. And the red series coloration has been used to allow us to observe collagen organization and orientation at the stretch mark level compared to the regular skin. So here we have the images of the skin explants treated with Sirius Red. We see the control, which is the first image on top, has a quite regular organization, whereas in the second image, and we should have it in the square, there is let's say more packed and disorganized collagen. The red is more intense there. So at the stretch mark level, the structure is disorganized. Nevertheless, still at the stretch mark level, the treatment with Centella Cast 0.5% for 5 days on the skin explants has allowed a massive collagen reorganization, so that in a way morphologically the dermis is far more similar to the non-stretch marked area. And the explants, beside the morphological appearance, have also been analyzed for fibers orientation. So collagen 1 fibers have been quantified at different degree angles, at 10 degrees intervals. Normally, we want to have all the orientation that is filled in by collagen so that we maintain a good function of elasticity and of the elastic fibers. So let's see the results. At the stretch mark levels, the fiber occupation decreases, and we see that here by 33%. But on the other side, in the treatment with Centella, actually the occupation of collagen fibers has increased by 49% in comparison to the stretch mark explants, and by 37% increase in comparison to the treatment with the placebo. So what is observed is that also Centella has been able to restore bundles formation. Let us see how. So we see here the, I would say, light blue parts are the voided part, meaning the fibers that are no longer present, the mature and bundles fibers of collagen that are no longer present. We see that both in the stretch mark as well as in the treatment with the placebo, we do have an increase, an abundance of this, I would say, voided spaces, and these are actually filled in by the treatment with Centella. Especially at the 20 and 60 degrees of orientation. So after having seen what happens to collagen and also to collagen bundles, there is another major actor of the ECM and of elasticity, which is elastin. So same donor, other explants were treated with Centella Cast, and elastin was evaluated. So the quantity of elastin was evaluated by immunofluorescence and quantified by image analysis. So long story short, elastin increased. So the stretch mark typically has less elastin, but this was actually boosted by the presence of Centella asiatica. And I know we all prefer images, so actually the green dots on the bottom part of the slide are actually representing elastin by immunofluorescence, and we can see that at the stretch mark level there is a majority of darkness, whereas there is more green at the stretch mark level with the Centella asiatica, meaning that Centella has boosted elastin production. So after having seen transcriptomic genes, scratch test, what happens on explants, we wanted to have, of course, the clinical confirmation whether Centella can represent a solution for skin repair in general, and also with a specificity on stretch marks. So let's quickly actually recall the main mechanisms that should be tackled to counteract stretch marks. So a stretch mark is a disruption in collagen organization. One target is definitely to restore the collagen organization in terms of both appropriate quantity of fibers and also to have an appropriate fibers orientation. The other point is a lack of the elastic function. So we want to aim for increasing the elasticity and, of course, cells proliferation. So all these mechanisms have been positively assessed in the preclinical studies. So just let's dive into the clinical studies where we want to aim at seeing an increased ameliorated organization of the extracellular matrix and also to impart more tensile strength to the skin. So 54 women with postpartum stretch marks enrolled in the study. Something that I actually missed to mention is that we tend to assign and to associate stretch marks to pregnancy and to women. Actually, it is not necessarily so, meaning that stretch marks are also present in men, and typically over 80% of teenagers do have stretch marks, because stretch marks appear when the skin grows too fast. And that is very typical, especially now when teenagers have their growth sprout and they grow very quickly, kind of overnight or in just few weeks. They gain a lot of weight and weight. So we did this study on postpartum women, but once again, let us not consider stretch marks as solely associated to pregnancy. The application was foreseen 3 times per day over 4 weeks, and the evaluated points were skin thickness, microcirculation, skin elasticity, together with an overall evaluation of the appearance of the stretch mark. So epidermal and dermal thickness as well as dermal density are actually important parameters to assess the cutaneous regeneration that is associated to the improvement of stretch mark. And we can see that especially at the center of the stretch mark, we had an improvement in skin thickness by 39%. Then, as far as concerning skin elasticity, it ameliorated by 62% in a statistically significant manner compared to the placebo, which actually has not been able to induce any statistically significant difference. Another characteristic of this lesion of stretch marks is that actually microcirculation is normally impaired. We can see that in the image. There are marked irregularities in both temperature and perfusion. So over the course of the 4 weeks, the microcirculation is normalized, and it has almost normalized, and it has increased by 162%. That was measured by laser Doppler flowmetry, which exactly measures the microcirculation. And that was measured at the stretch mark level. So finally, as I mentioned, there has been a self-assessment that was requested to volunteers to evaluate on a visual analog scale how they rated and they perceived the appearance of their stretch mark, and they rated an amelioration by 42%. So by 42%, their stretch marks were less visible. And so the biological characteristics of the formation of a stretch mark imply, as we have seen, different mechanisms. And Centella has been reported to act on all these mechanisms, like promoting collagen organization, increasing the production of elastin, replenishing fibers orientation, and promoting collagen bundles formation, promoting microcirculation, increasing overall skin elasticity and thickness, and finally also ameliorating the visual appearance of stretch marks, which are in a way mitigated and erased. So just in a nutshell, that's all we have just gone through. I am being conscious of time. So just in particular, a reminder, this Centella asiatica is not just a general extract of Centella. It is combined, highly pure. It's the combination of a fractionation of highly purified molecules from a pharma-like blend, from a pharmaceutical legacy. So there are some topical pharmaceutical ingredients that would also have a very similar version of Centella asiatica. Some technical information, the product is a powder. It can be dissolved with, I would say, quite a handy procedure. I would like to draw your attention on two things. The first is the INCI name. So we do not have the general INCI name because we are dealing with purified molecules. So the INCI would be asiaticoside, asiatic acid, and malic acid. And also I would draw your attention to the logo, the green logo at the bottom of the slide, which is called IDPAC. It means that aside of all the incoming controls that we are doing on Centella asiatica, meaning the botanical control, chemical microscopical control, HPLC for markers and all of that, also genomic identification is carried on the plant, the incoming dried plant prior to the extraction and diffractionation. So key learnings, the benefits of Centella can be summarized in promoting dermis reorganization. And this actually, of course, applies not only to stretch marks or to skin repair, but as we have been seeing at the beginning of the presentation, it actually applies to different uses. It may be used for hair or scalp care, for instance, because it promotes microcirculation, and also it improves the hair follicle health. It may be used in facial care as anti-aging and anti-wrinkle because it actually deeply reorganizes the dermis. It may be used in lips care, both for the potential dermis reorganization as well as on chapped lips to promote skin repair. It may be used in body care as an adjuvant to slimming, in promoting microcirculation and also skin elasticity and firmness. It can be exploited in legs wellness products. So skin repair properties may actually be considered as, I would say, a very general starting point, then can be used in many different cosmetic formulations. So we have 1 minute left. It was a rush.
Thank you,
Thank you.
Giada. That was a really good, informative presentation. I think we see what you mean with regards to pharmaceutical-like. So, you know, we always have to be careful in the cosmetic regulation industry to keep our claims quite conservative. But the product, it looks, due to your efficacy data there, like it is performing very well. So we have a question and also a comment. The question is, you mentioned oral care. So the question is it advisable, I mean, the applications across oral care, so toothpaste, mouthwash, because we know like ginger, bisabolol, they're used for soothing of the gums, soothing mouthwash, soothing toothpaste. So is it suitable, since it's in a powder form, is it suitable across all these oral care applications?
Oh, yes, I would say yes. There is a procedure, it's in a powder form, and nevertheless there is a procedure to solubilize it. Of course, in a toothpaste, it's a little bit easier. A toothpaste is very rich in powders too. In mouthwash, there might be some alcohol. Of course, this actually very much depends. The oral care application was, in a way, I would say a consumer's wish from Chinese consumers. It was more of a, I would say, consumers insight study where Chinese consumers would like to see Centella asiatica in their oral care products. Definitely, to me, what makes sense is for the sensitive gums, for bleeding gums, Centella asiatica makes sense in the reorganization of the connective tissue that gums are made of.
Perfect. And then a comment, Giada, is that as women naturally, you mentioned in your presentation that men also have stretch marks, which we are aware of. We don't see it as much as women, but so you mentioned you were speaking about the reorganization of the collagen and the efficacy study. So with the stretch mark that we're finding often, it gets almost like when it gets that silvery white appearance, it's very, very, very difficult to actually eradicate. You can't eradicate it, first of all, and second of all, it's very difficult not to still notice the stretch mark. So my not a question, but it's just a comment, is that we see all the efficacy data, but do you also agree that particularly after a lady is a certain age and you've had that stretch mark for a long time, when it's not a fresh stretch mark, so a lot of the time the companies, the brand manufacturers, have to actually do a lot of preventative, like you get preventive aging, you're almost using these type of applications before the stretch mark is very advanced. So I've noticed with also with African skin compared to white skin, once that stretch mark is in place for a good few years, it's really, really, it gets almost like a certain color to it. Whether you're black skin or whether you white skin, it gets a silvery look to it. So it becomes quite difficult.
Yes, definitely. So what I actually failed to let's say indicate is that the volunteers of the clinical study were moms at their second after their second delivery. So they already have had two babies. So they had a mix of, I would say, kind of fresh stretch marks, so the red ones, and also they had more, I would say, stabilized and white stretch marks from their first pregnancy. Definitely, stretch marks are more difficult to erase, when they are white, when they are the so-called white striae. So definitely that is true, but that is actually that was part of our study. So we did not actually discriminate between the fresh stretch marks and the older stretch marks, but there were older stretch marks as well. And from what I am understanding from the data, Centella Asiatica is actually largely working on skin repair and reorganization, no matter how long that skin has been disorganized in the past.
Okay, perfect. Thank you for that elaboration, because it's quite an important point to take notice, because often we get excited, even as manufacturers of raw materials or representatives of raw materials, and it's actually a lot like preventative medication. It's preventative aging, it's preventative. You actually have to start applying some of these things on earlier than later, when more of the damage is done, to get the best results. But your elaboration of your test results is very informative. Thank you for that.
Thank you for the question and the comments that were very insightful too.
Okay, Giada, thank you very much for joining us all the way from Italy. We hope to see you again soon, and very informative. Always nice to hear about how you fractionized and done all the technology on these raw materials, and anything that's giving extra efficacy, we're always welcome to hear about. So we're going to be moving. Thank you very